Bismillahirrahmanirrahim. Today in this video lecture we are going to see HBeAg and HBeAb. So we are going to diagnose the HBeAg and HBeAb through ELISA test. So let's start the video. So what is HBeAg and HBeAb test means? So it is actually the diagnostic test that is performed by ELISA, and in which we find out the antigen of the hepatitis B virus and antibody that is developed against the hepatitis B virus. Okay, so this is a little bit about HBeAg and HBeAb test. Now let's talk about the HBeAg. So HB means hepatitis B, E means the antigenic portion that we call that envelop, Ag means antigen. So HBeAg means hepatitis B virus envelop antigen. So when this antigen, we better note that the virus structure contains the envelope portion. So when the envelope portion or the antigen portion that is present into the bloodstream, and when it is diagnosed through ELISA test, so it means that the patient have chronic hepatitis B virus infection, and it also means that the hepatitis B virus are in the replication form. Okay, so this is a little bit about the HBeAg. Next we have HBeAb. So HBeAb means hepatitis B virus envelop antibody. So when the antibody is developed against hepatitis B virus, we call that HBeAb. So when it is diagnosed through ELISA test, so it means that the patient are recovered from the acute hepatitis B virus infection. So what is the main purpose of HBeAg and AB? So the main purpose of HBeAg and AB is that to find out the treatment that we are providing for the hepatitis B virus, either the treatment is workable or not. So this is the main purpose of HBeAg and AB. Another purpose of HBeAg is to find out the chronic and the acute stage of the hepatitis B virus. And the HBeAb, the main purpose of HBeAb means to find out the antibody that is developed against the hepatitis B virus infection. Okay, so this is a little bit about the purpose of HBeAg and AB. So now let's see the requirements of HBeAg and AB test. So for this test we need the patient blood into a red cap, or we also take it into the gel tube as well. We need a HBeAg and AB kit, as you can see. So this is Ag and AB ELISA kit. So this is the HBeAb ELISA kit, and this is the Ag ELISA kit. Now let's see that what this kit contains. So first of all we have ELISA HBeAb kit. So this contains the wells, as you can see. Now this is the wells of HBeAb. This well is coated with the antigen. So when antibody is present into the blood, so it will be detected. This kit also contains the conjugate. So this is the conjugate. It also contains positive control as well as the negative control. Moreover, this kit also contains the stop solution. So this is the stop solution. Now this kit also contains the color A solution. This is the color A, the white one is color A, and the black one is color B. So the same thing is also present into HBeAg kit as well. So it also contains this wells, as you can see. So this is the wells that is coated with the antibody. So when antigen is present, so it will be reacted. So this is the HBeAg wells. We need a stop solution. This is the HBeAg conjugate. So this is the conjugate. and it also contains the positive as well as negative control. So let me show. and it also contains the wash buffer as well. So wash buffer is also present in case of HBeAg and HBeAb. So this is a little bit about kit. Now we also need yellow tips, we need pipette or jester. So now let's see the procedure of this test. So first of all, we should centrifuge this blood to get the serum, and we'll place the tubes to each other, as you can see, to make the balance equal. Okay, now then we on the centrifuge, and we'll adjust it for 1000 rpm for 1 minute to get the serum. Once the centrifugation is done, so we get the serum. As you can see, this yellow one is serum and this red one is the RBC. Next we should take the wells. Okay, now this upper portion is for the AB and the lower portion is for the AG. So we should take the wells and we'll place it into the ELISA plate. So this is the ELISA plate. Now this first well as we are taken, so it is the test. Okay, now we should also take the positive control and the negative controls. It means that we should take 2 more wells for HBeAg and HBeAb respectively. So the second well that we are taken for HBeAg and AB, it should be act as a positive control, while the third well that we are taken for HBeAg and AB respectively, it should be act as a negative control. So in the second well of HBeAg and HBeAb, we should add positive control, and in the third well of HBeAg and HBeAb, we should add negative control. Okay, so we can also say that the upper row as we are making it is the HBeAb, and the second row, or this is the second row, so it is the HBeAg. So next we should add conjugate. So we should add HBeAg conjugate into Ag portion, that is the lower portion, and then we should add AB conjugate to HBeAb portion, that is the upper portion. So we should add 1 drop of HBeAg conjugate. Okay, now this HBeAg conjugate that we are added in each of the wells respectively, this conjugate contain an enzyme which we call that HRP. HRP means horse radish peroxidase. This enzyme is used for the color, to provide the color, or it is used for the color. Okay, this conjugate is also used to combine the antigen and antibody complex. After that we should take 50 microliter of the blood serum. Okay, and we'll add it into the first well of the HBeAg and HBeAb that is acting as a test. Next, in the second well of HBeAg and HBeAb, we should add the positive control. So we should add 50 microliter of the positive control in HBeAg and AB of the second well. Positive and control it will give the positive reaction, and the negative control will give the negative reaction. Similarly, in the third well we should add the negative control. Okay, so when the positive and the negative control are well, and when we get positive as a positive control and negative as a negative control, it means that the kit are okay and there is no problem, and the test that we are performed, it is also okay. Okay, so this is the importance of the positive control and the negative control that we should perform with the test. We can also say that this positive control or negative control contain the noun sample, means positive will have noun positive sample and the negative have noun negative sample. Now once we add this, so then what should we do? We have to incubate it. Okay, so we should incubate it for almost 30 minutes at 37 degrees Celsius. So this is the incubator. Now we should place it into the incubator. After 30-minute incubation at 37 degrees Celsius, what should we do? We have to wash it. So now the incubation process is done. So after 30-minute incubation, we will take it and then we should wash it with the wash buffer. Now in this case, we should make 20x wash buffer. So the wash buffer of HBe, Ag and Ab is very important, because it will wash out the remaining antigen and antibody that is not bounded with the wells that contain antigen and antibody respectively. So this wash buffer, as you can see, we should make it by adding 1 ml of the wash buffer solution and 20 ml of the distilled water. Okay, so this is how to make 20x wash buffer for HBe, Ag and Ab. So once we take the wash buffer as we already make, and then we should have to fill it with this. First of all, we should discard the solution or the the liquid which we added the mixture, and then we add the wash buffer. As you can see, we will a little bit mix it and then we'll discard it, as you can see. So we should have to wash all these wells 5 times. Okay, so the washing process will be 5 times. So now after that we should dry each well. So these are the wells, so we should have to dry it. Okay, now we will place the tissue paper and then we'll will invert the ELISA reader plate, as you can see. So what will happen, the the wells will be dry. As you can see, the wells are now dry. Next, what should we do? We have to add the chromogen solution. So first of all, we should add color A solution in all these wells, as you can see. Okay, so what is the purpose of color A? So color A is we can say TMB. It is actually the substrate that provide a bluish color, that is the positive reaction, and color B is providing the yellow color, that is the positive reaction, and it is actually OPD also. It is actually the OPD, and color A is the TMB. The main purpose of color A and color B is to detect the antigen antibody complex. It also visualize the reaction, and it also quantify the antigen and antibody, etc. So after the adding color A and color B, 1 drop in each of the wells, so then we incubate it in a dark environment for almost 15 minutes. Now after 15-minute dark incubation, then what should we do? We have to take it and then we observe the color, as you can see. So in the first well of Ag and Ab, it is the colorless. It means that it is the it is the negative. So after that, what should we do? We have to add a stop solution. The purpose of the stop solution means we should add 100 microliter of the stop solution. So the main purpose of this stop solution is to stop all the reaction, enzyme and substrate reaction, and it can also stop the over enhancement, etc. And it can also ready or make the ELISA for reading as well. So this is the main purpose of the stop solution. As you can see, the second well of Ag and Ab, Ag, the second well is the yellow color, and in case of the Ab it is also yellow color, but in case of the Ag it is positive, and in case of the Ab it is negative. Okay, the yellow color is negative in case of the Ab, and it is positive in case of the Ag. Similarly, in case of the Ab, the negative is yellow color, and in case of the Ag, the negative is the colorless. Okay, so don't be confused, it is actually opposite to each other. Now, how to read? First of all, we should place the ELISA reader plate into this machine, as you can see, and then we should read the test. So what will we do? We will click it and then we will click on to the rapid, because for AB we should click on the rapid, and then we will arrange all the things, means we will set the wavelength, we will set the normal range. So more than 1 it is negative, less than 1 it is positive. Okay, remember, in case of the AB. So then once we arrange it, so then we will click on the serum, and then what should we do? We will click and will select the wells where we put the sample. Okay, so now as you can see, this second row, we have selected the second row because in the second row we placed our sample of the AB, and then we'll click on the test, and then it will read it. After a few seconds we will get the result. In case of the AG, we will click on the single and will click the test, and then it will give us the reading of the AG. So we get the AG and AB reading, as you can see. Now let's understand ourselves about this result. So this fifth row, as you can see, I think the fifth row, this is the AG. Okay, this is the result of the AG. So the first well is actually, we get the less than 1, so it means that it is negative. And then the second well is actually positive because we get more than 1 value, means 13, it means that it is the positive. And the third well, it is also negative, it means less than 1, so it is also negative. Then we have AB. So this, the second row is actually the AB. So the first well is actually negative because it have more than 1 value. The second well is also negative because it is also more than 1 value, and the third is actually the positive because it is less than 1. So you can also predict the result by this, the color as well. Okay, as you can see. So this is all about the HBeAg and HBeAb ELISA test. So I hope this video is helpful for you. So please like this video, share this video, and also subscribe the channel. Thank you so much for watching.